Our next speaker is Jiayun from University of Greenwich, and will tell us about natural language processing-based deep learning approaches to predict solubility parameters.
Thank you. So my research background is actually in kind of using traditional molecular dynamics simulations and quantum mechanics calculations to understand molecular properties and to understand organic reactions and enzyme catalyzed reactions. But since last year, I've started to look into how deep learning approaches can be used to understand the type of research questions that I've been working on. And of course, machine learning has been around for a long time. But I think it's only in the past few years that we started to see the impact of machine learning in our life. Google Translate is very accurate now. I'm amazed how accurate it is sometimes translate between different languages. And Alexa and Siri is all around. My son's 3 years old, and he can ask Alexa to add his favorite snack to the shopping list. My daughter is 7 years old. She will ask more philosophical questions like, Alexa, do you think Santa is real? So I think their generation, they are growing up with this kind of virtual assistant, just like our generation was growing up with PC and the internet. So I think the real life impact we're seeing from machine learning is mainly down to the development of the computing power and also some of the state-of-the-art deep learning algorithms. And there's many ongoing efforts to try to see how we can use deep learning and data-driven approaches in the broad field of biology and chemistry. And then there's some success. For example, Google's AlphaFold, and I think I didn't quite like the way they've done the marketing, say it will change everything. Maybe it generates lots of structures. But from structures, we use that to eventually go and discover life-changing disease treatment and many other real-life applications. There are many steps to go from there to that, where we are with like Alexa and Siri. So I think we are yet to see the full impact of deep learning and how they can be applied in our research field generally in chemistry and biology. And so there's a lot of opportunities. And when I spoke to my collaborators in kind of machine learning and natural language processing, and they would sometimes comment, oh, you are like us in 10 years ago. So I think we are at this stage that everyone is trying to see how we can use deep learning, but there are many ongoing open questions that we can study and explore. And then there's a lot of opportunities both in terms of research and in terms of commercialization. So deep learning, of course, is very data hungry. So we need big data or lots of data. And we think about the kind of big data we have in biology and chemistry. We have a lot of sequence data, protein sequence, DNA sequence, and so on and so on. And we have some kind of database for molecules or proteins with their functions. So that's kind of providing label for training. And we have, of course, structure data. And we have huge data of chemical structures in a range of billions, and then some with a small proportion with properties, and in well-established databases like PubChem and Zinc. So if we look at those type of big data available in our research field, then really one type is sequences, and that has strong similarity to kind of language sequences. And we look at structure data that has similarity with image. So this opens up opportunity for us to integrate techniques or concepts from other domains of machine learning, for example, natural language processing or computer vision, to study what we have in our data set. And it's interesting, actually, to see many of the tech companies are going into our research field. So nowadays, it's not a surprise for me to do a search and find Microsoft publish something in something I'm interested, because those tech companies are very good with their deep learning algorithms, and for them, it's really just to adapt their algorithms and trainings to a set of data from different domains. Okay, so to convert a molecule for machine learning, there are different ways, and that's just a very simple example of a simple molecule. And one way that is we convert it into a three-dimensional structure, and from that we can use image recognition machine learning approaches, kind of truncate that structure into different frames and then process those as images. And then we can use QM-based approaches, and we can derive features from that. I think the first speaker mentioned the kind of features that we can usually compute. They come from QM calculations, so like shape and size, electrostatics, and molecular orbitals, and so on. So another direction is that we can convert them into text. And then, many like smiles, the first speaker mentioned about smiles, but there are a few others like selfies and so on. But they all look like in this type of structure. And the next step is how we break them into substructures. So I think you can also call them tokens. So very simple, you break them into one fragment, two fragments, so on, until your fragments cover all the structures for your smiles. And then the next big question is how you convert those substructures into mathematical vectors for machine learning. And there are various ways to do it. So one is kind of quite a conventional approach, one-hot encoding, that we have, for example, 5 tokens in a smile or in your whole database. And then you just give them a positioning label, and you can use it. It's very simple, but actually very effective in some of the research questions. Another approach is the more traditional, widely used molecular fingerprints. So we're looking at, depends on how you define the radius that you can break your structures into fragments, like from typically between 1 to 3 bond length for fragments, and you kind of encode those into you typically 1K to 2K bit vector. So this vector represents your molecular fingerprints. But there are other ways to try to generate the vectors that are more compact and more information rich. So because smiles has very strong resemblance to text, therefore we can borrow some of the approaches from natural language processing. And then NLP is a branch of machine learning that is to process and analyze text. And there are some breakthrough developments in recent years in NLP, and that leads to many applications that we use. For example, when you write email, and then your server would automatically predict what you're going to write next. I think that's building on something called transformer in NLP. And so through smiles, it's possible for us to use kind of the NLP algorithms to analyze chemical structure in a similar fashion as they are used to analyze text. So when I started to work on this last year, I started with a model called word-to-vec. It's actually considered a dated approach in NLP now, although it was only developed in 2013. So that's how fast the field has moved. So in this approach, I think it's quite interesting to use this as a starting point for me to understand, because as a newcomer last year, there was a very steep hill for me to climb to understand the NLP approach. So word-to-vec was a good starting point for me. So this model trained on usually a large corpus of text, maybe the whole Wikipedia or something, to determine the vectors that represent words. And those vectors are determined by the context the words are used. So if we look at this example here, we have 3 words. We have king, man, and women. And there's some similarity between king and a man. So this is a illustration of the vector for each of the word. And similarity between king and man, but difference in one dimension of the vector because, of course, king has additional meaning in the word. And if you look at man and woman, again, similarity in its vector, sum of dimensions, but difference in that to explain the difference between the 2 words. And if we plot this in the vector space, and we can see kind of king and queen would occupy similar space, and a man and a woman would occupy similar space. But the distance between those 2 are similar, because that's the difference between between king and a man, queen and a woman. So that's this kind of model. I think we can use it to try to understand chemical structures. So we can tokenize our smiles in similar ways. So we have substructures. And then by training them, going through a word-to-vec model, then we can capture, hopefully, the meaning of the fragments and the chemical fragments and how they are related in mathematical space. So what we've done is we trained a word-to-vec model using about 2 million smiles, a relatively small representation, but they are all biological active molecules. And so each common molecular fragments will have been assigned a vector called embedding in NLP. And so where their chemical properties related to other fragments are kind of embedded in those vectors. And then now, whenever we have a new molecule, this molecule can be represented by a matrix formed by vectors corresponding to its fragments. And this matrix, sometimes you can use a matrix for different machine learning tasks, but you can also combine it to form into a one-dimensional vector. And they can be used for trainings. So just to give you an idea, so if you have, again, very similar molecules, and then if we represent this molecule in a matrix of vectors, and that's what you usually have. And the dimension of that usually will be hundreds of vectors, hundreds of dimensions. So the kind of labeled data that I'm interested in is called solubility parameters. So it's a very widely used parameter to help us determine various tasks in drug discovery and in pharmaceutical formulation. There are various parameters that we can use to understand solubility. And the ones that I was interested in was called Hansen solubility parameters that has 3 components to explain solubility of a molecule. So we have the dispersion forces, we have the hydrogen bonds, and we have polarity. And the 3 of them, and if you have those parameters for each molecule, and there are ways to calculate, very simple mathematical ways to calculate the distances between, so they are like Cartesian coordinates. And then you have 2 molecules, and if you calculate the distance between those Cartesian coordinates defined by the Hansen solubility parameters, then you can roughly estimate they will be soluble or not. So that's kind of the data set I'm interested. So we had a data set of 1,200 organic molecules, and they all have their experimentally determined Hansen solubility parameters. And then we kind of assign their molecular embeddings from the pre-trained word-to-vec model, and we split the data for training, validation, and testing. And here are just some examples from the molecules that we've studied, very simple organic molecules. So the kind of, we've used a range of models to see how best of a prediction of the Hansen solubility parameters are based on the embeddings. I think to benchmark, we use kind of a feature-based model. So we have, the features are constructed based on molecular fingerprints, charge densities, electrostatics, and shape and size, and then that forms one vector and we use it for machine learning, and I think that's a linear machine learning model, and then the R square for the predicted solubility and the experimental determined solubility is around 0.77. And then we've done a few methods for including linear, simple FFNN type of neural network, and we've used RNN, and to see how our prediction would compare with the discrete feature-based approach. So I think in general, those models are not bad, considering we haven't actually spent weeks to compute those features. So we got those features based on just simply learning from a bigger data set. But the performance of that are slightly below kind of the feature-based approach. So what we are going to, what we're working on is, sorry, how long do I have now? 5 minutes. What we're working on is to use a more state-of-the-art model. As now, once I got into the field, I realized there are other better models than Word2Vec. So the limitation of Word2Vec is a static embedding. That means that each of the vectors representing those mathematical, the substructures or tokens from smiles, they are static. And they don't change when this structure is used in a different molecule. And that's obviously not accurate for chemistry, because we know even it's the same functional group, if they are used in different structures, their properties will be different. So I think there's intrinsic limit in terms of the static embeddings from this model. And then the more state-of-the-art models at the moment is transformer-based model, and that was developed in 2018. So related to transformers, we know like models like BERT and GPT-3. So the benefit of transformers is that it's non-sequential and in a sense better of the weighings of the different tokens in the sequence. And then we can update those weighings of the tokens of the vectors by from pre-training in fine-tuning in the second step. So there are some initial models, for example, CAN-BERTA, and then kind of that has been trained on 77 million smiles and kind of have a broad understanding of the chemical space. And then, of course, that we can train similar models based on BERT, but with our own different training strategies and with additional learning strategies. So just to explain the difference between static embedding and contextual embedding. So if we look at the words of mouse and as the animal or as in computer, so static embedding, as like in the Word2Vec approach, is just two vectors showing the two different meanings of one word. And then if it's contextual embedding, as in BERT, then really we define mouse in kind of cheese-loving mouse or fast-moving mouse or click on the mouse. So we give those words more context of how they are used. So I think this will be extremely useful when we look at chemistry, because we know, like in smiles, that the distance of tokens in the smiles, they do not usually correlate with the space they have in three-dimensional structure. And also distal tokens sometimes would very often have a strong influence on each other's chemical property. And therefore, I think using similar approach as to transformer and BERT may introduce some benefit in terms of the questions that we're interested. Just to summarize, I think at the moment, whenever I go to talk to colleagues in drug discovery, and they'll say, oh, I know, kind of, transfer learning and embedding, but are they good enough? Are they beating the state of the art? Why do we need it? So at the moment, I think the embedding approach or the kind of the bigger language model transfer learning is still not exceeding the traditional feature-based model. No, we're not there yet, but there are various things that we can do to improve the training of the model. So water-vec is only very, well, not very early by our standard, but by NLP standard, it's an early and simple model. And we're working on more state-of-the-art deep learning models. And it's, I think the things I'm talking about today, they fall into broadly the transfer learning. So it's in terms of learning from larger data set and then transfer that knowledge into a smaller set of data with more labels. And that can be quite useful for chemistry, because what we have is almost unlimited chemical space defined by those hypothetical molecules. I think it's in the region of maybe not billions. And how we can learn more from that, this chemical space, and transfer those learning, and then with limited label data we can generate either from experiment or from QM calculation. I think that's the challenge. And there are techniques that we can use, for example, contrastive learning, which is a technique that we can really learn more from those unlabeled data. And there are wide applications of this, of course, and we can apply other data sets, such as chemical reactions and protein sequences with similar approaches. And acknowledgement, thanks to the university's HPC facilities for the GPU, and my project students, who were brave enough to learn programming and machine learning from zero background as a chemistry student, and my collaborators in NLP. And this is a picture of the AI, when AI was asked to draw a sea otter in the style of a girl with a pearl earring, and that's what it came up with. Quite interesting. Captures the sense, but yeah. And this is just some news from a few weeks ago that Meta has a kind of large language model that I've mentioned, and it didn't survive for more than three days online because of inaccurate in its content and problematic content. Yeah. So just remind us that AI is there, but it's for us to determine, to validate it, and to control it. Thank you.
Thank you. Questions? Yep.
Hi, thank you for the really interesting talk. I was just a bit confused when you discussed the matrix representation of molecules in the Word2Vec, because you said you can add up the columns to get a 1D vector, and I was wondering, how does that retain enough information about the molecule?
No, it's not as, so you can, so for example, let's go back to, yeah, I think it's this one. Ideally, you can use the full matrix with the sequence. So for RNN model, for example, you have to go to input in RNN model in this sequence. So you you preserve the sequence information of it. But for others, for example, like if you just want to do a linear model or FFNM, sometimes having that is not essential. You can combine them, just sum it or do an average of it.
Other questions? Okay, I have a question. If you go back to the slide where you show the results.
The results?
Back.
Yeah.
This one. Okay, so you calculate using DFT the properties of the molecules, but DFT, there are many flavors with various degrees of correctness in predicting the results. Did you investigate the effect of on the quality of the fitting based on the quality of the DFT model or other non-DFT models?
No, and I think we just use some very low level of DFT to compute those descriptors. And I think actually, it's, if you look at the contributions of those features into the model, it's the simple ones like shape and size that contributes predominant to the information that the model has learned. So in this way, I think charge density is probably not in the middle range, but shape and size and really basic information about the molecules are quite important for this task. So I guess in this, if the electrostatics are not that important, and then is it worth investigating a lot more time to use really high level of DFT to calculate them? So yeah, I guess at the end it's a group representation of the molecule that's important.
Okay, James?
So I work with a lot of pharma people all the time, and they tell me that charge is one of the most important features in drug discovery.
Yeah, I guess it depends on the target, the prediction target. And I think I didn't show that in this. Because I have three targets. There is the hydrogen component, dipole, and also there's polar, I think. So it depends on which target you go for. The contributions of those descriptors will be different. I think for simple ones, so I think hydrogen usually performs best for hydrogen component, and then for that shape and size, and then charge will be quite effective. But I think the model performs really poorly with the dipole, and I think, with the electrostatics, maybe there's ways that we can improve the descriptor based. But that's not my main research, because I want to really look at different ways of representing molecules to make it at least as accurate as the descriptor based, because those takes weeks to compute. When you have thousands or tens of thousands of molecules, you need to have extensive computing of them.
Okay, if there are no more questions, let's thank our speaker again.